Hello, my name's Elise, and I will be recording the flow cytometry for Module 8 of Immunology. So what is flow cytometry? It is the use of lasers to measure size, number, complexity, and what may be bound to cell surfaces on like a series of cells that are passing through a stream of saline, basically. I can measure things from 0.5 to 100 microns in size, but typically it's used for things that are 1 to 14 microns in size. It can go bigger or smaller depending on what type of machine and reagents you're using. So the cells are in solution and they are directed or shot past a laser in a stream of saline or some other non-reflecting solution. So this is just like a screenshot from a really helpful video that I found. So as the cells pass the laser, the light is directed either through the cells or scattered by what's inside the cells. And so what passes through is called forward scatter, and that's determined by like a minimal angle of scatter right here. So it's not exactly the same, but it's beneath a certain threshold angle. Side scatter is what is reflected in every other direction other than straight forward. Okay, and so by recording the forward scatter, you're able to determine how big the cell is. So the more forward scatter you have, the bigger the cell, and the side scatter measures the complexity of what's inside the cell. So the more side scatter, the more complex whatever the cell is that you're looking at. So these two metrics or dimensions kind of measure the forward scatter and the side scatter. They measure things that are then plotted basically on the same two-dimensional plot, so you can kind of understand how big things are and how complex they are in the same graph. So in terms of if you were just looking at forward scatter, you would be able to tell. You kind of make a histogram based on how many data points you get of a certain size, and then you kind of make a histogram of how frequent they happen. And then for the side scatter, the same thing. You get data points based on how much side scatter there is. So these data points that I've been talking about, they are so when the laser passes through, there is like a photoreceptor on the other side of the wall. And it's not measuring the light that the laser itself, but it's measuring what's scattered. So there's actually something to kind of stop the laser itself, and it's only measuring the scatter. So the photoreactive sensor is measuring the photons and the light that hits the sensor, and it turns it into voltage. So every time that there is like kind of a ping of a photon against the sensor, it sends an electrical impulse, and that's what the computer reads. Alright. Okay. So histogram can be made from each dimension, the forward and the side scatter, to plot the quantity and size of the cells in the sample, and you can plot these two graphs, these two like values, two-dimensionally to understand what type of cells are in your sample. I found a really great clip. It's easier to just show. So this is kind of just saying how the two graphs are plotted in two-dimension to form like this type of looking graph, where you'll be able to see this right here, which is like one representation of a type of cell, and like this might be another, and this one right here would be a third. Okay, so the next part would be like when they were asking about the fluorescence activated cell sorter. So that is when you get wee little fluorescent molecules like antibodies or something, fluorescent antibodies, and you tag certain cells with it. And by doing that, you kind of just introduce the antibody or the molecule into the sample before it goes through the interaction point. And so it could happen, the antibody or the molecule could bind on the inside of the cell or on the outside of the cell, and it could happen once or it could happen many times. So when it passes through the point of the laser, and the laser hits the fluorescent molecule, so the wavelength of the laser that hits is going to be different than the wavelength of the emission that comes from the fluorescent marker. So let's say you have like a green, something that fluoresces a green wavelength, as your cell is passing through and it hits the laser hits the green fluorescing molecule, there can be sensors that kind of detect like, oh, there's something green on that cell. So this is useful because you can tell, one, if there's like more than one receptor or more than one fluorescent molecule on the cell, you could be able to tell that there is more than one based on the intensity of what the fluoresced like wavelength was. So like let's say it was like, yeah, it was green, but like this cell had a lot more green, this cell had a lot less green. It could also be used with more than one color. I saw something that was like up to like 8 different colors at once. And that just works by like you go through like different prisms of like what type of, or what color, what wavelength is going to be read on which type of like photoreceptor further down the line. But it's all based on the same type of principle where there's, one stream of cells getting passed through a laser, and the laser hits either part of the cell and it scatters, or it hits a fluorescing molecule and then it like sends out that green emission for whatever color. And that green emission, or the side scatter or forward scatter, is picked up by a photoreceptor and then turned into voltage or some like electrical impulse, and that's read and plotted so you can better understand the size, quantity, complexity of what cells you're looking at, or that were in the solution. So in terms of applications, this could be for immunophenotyping, which is identification of the markers on cells. It can be used to just identify a single marker or more complex identification, and you would have to use like a homing profile, activation states, cytokine release to do that more complex identification. You can also use it to diagnose diseases or monitor and evaluate residual disease, and then all these other like apoptosis, cell proliferation measurement, and cell cycle determination, small particle detection, gene expression and transfection, and just other uses in immunology. Apparently, it's not as as popular in other fields of biology and microbiology, but immunology is like they rely very heavily on cytometry, which I guess was interesting. And that is the end of my presentation. I hope it made sense and that you could hear me.